Okay, well, thank you very much for inviting me. My name is Carlos Toscano, and I'm a PhD student, but actually it will take like less than 1 month or 1 month and a half to earn my PhD degree, because I have finished my thesis and I only have to defend it. And well, I am a biologist. I did the licenciatura, which is a 5 year degree. The old system. No, it's another system in biology, and then I did it in Sevilla, and then I moved to Barcelona to do my master in biomedical research, and then I started my PhD. Officially, the PhD program is biomedicine, but my PhD is more focused in theoretical aspects of biological computation. That's it.
That's a really good explanation. Okay, moving on to you, Jaume.
Okay, so I'm Jaume Puig. I'm a biomedical engineering undergraduate, and I'm doing my bachelor's thesis at the Translational Synthetic Biology Lab at the PRBB. And I'm also a member of the Open Science and Accessibility Committee for After iGEM, because I participated with iGEM in 2020 and decided to enroll in this committee. And that's all.
So I see a lot of synthetic biology. Great. So moving on to the last guest, Juanma.
Okay, hello. First of all, thanks for inviting me. Well, I'm a 4-year student of biomedical engineering also. And yeah, I'm also studying physics now. So I'm like studying both careers at the same time. And well, I am developing also my TFG in the Integrative Nanomaterials Lab. So I'm working with nanotechnology and all this kind of stuff. And well, that's it.
From my personal standpoint, synthetic biology is using biological tools to develop new biological systems and or devices to a very vast variety of uses that can reach from medicine to maybe industrial applications. In fact, I think a good explanation for what synthetic biology stands for is maybe the quote from Richard Feynman, which every synthetic biologist has heard. That is, what I cannot create, I don't understand, which interpretation is that only by designing and building these biological systems, we can truly understand the principles behind them.
Okay, synthetic biology. Well, but I would like to point out a couple of things. First of all, synthetic biology, the idea of synthetic biology is not new. And when I say that it's not new, I'm not saying that it starts with the technology of being able to modify or alter DNA. Okay, first time that synthetic biology was conceived or explicitly mentioned was in a book by a French scientist, Stéphane Leduc, in 1910, okay, 1910, and maybe it's possible that some of you may expect that the idea that he had was very different from our current conception of what synthetic biology is. That's not the case. That's not the case, which is very surprising. A guy from the very beginning of the 20th century aiming that synthetic biology would be the ability to create living systems. He said organisms, but it's systems, living systems with the ability to carry some specific function, and the idea would be that one of the main ideas would be that you are going to be adding several functions in incremental steps. Like, you are not going to create everything from the ground up, but or from the sketch. But okay, let's first create this tiny system with a specific task, and then use that for creating or to implement another more complex systems. And I find fascinating the fact that it's been a century, and actually more than a century since this idea appeared, and now we are just doing this. I think synthetic biology has two main branches. One of them is about we want to create systems with the ability to do whatever we want independently. Another thing is if this functionality is, has some kind of commercial utility or not.
That's where
The point
IDEM
is that
comes, I think, because you can do whatever you want.
exactly. So if you're implementing something new, you are doing synthetic biology. And another branch of synthetic biology is the standardization part, right? The biobrick and all that stuff.
Well, my vision does not differ much from the definition that Carlos and Jaume have given before. So, in my opinion, synthetic biology is defined as the synthesis of biomolecules, or maybe even the development of biological systems with new functionalities that are not a priori found in nature. And well, in my case, I developed my TFG in the field of nanotechnology. And to be honest, I strongly believe that the development of this area is very important for the advancement in synthetic biology and in science in general. I don't know, thanks to nanoscience or nanotechnology, we have new materials and also new tools that clearly expand the horizons of synthetic biology and science in general. So, well, I think that, to be honest, and this is important, I think it's remarkable. I think that it is essential to understand that multidisciplinarity and especially collaboration between scientific fields is key for scientific progress. This is at least my point of view.
Let's move on to the second part, maybe of the live, which is more a discussion about several topics. So, for example, we can talk about bachelor thesis, or in the case of Carlos, PhD. So, what are you currently developing or have developed? Like a short summary of that. So, Carlos, could you give us a short explanation on what are you developing?
Okay. Well, I've been obsessed for years now with distributed computation. The idea of implementing digital circuits with logic gates within one cell, it has its limitations. First, because you cannot put everything you want inside a cell. Second, because in the case that you can put all the genes, the energy consumption is going to compete and win against the cell, right? So around 10 years ago, it appeared an idea of splitting your circuits among different strains that would communicate using little diffusible molecules like lactones used in quorum sensing. And I've been obsessed with the implications of chemical wiring, which is that idea. It is widely used. It's very common to see projects based on splitting quorum sensing genetic systems, which is not very complex. It's just only you put a gene responsible for creating and producing that tiny communication molecule, another gene responsible for sensing that molecule and induce gene expression of whatever you want. So you put these two systems in different strains, and this is very ubiquitous, and you can find it everywhere. But no one treated the implications of using that tool. It has implications. So if you want to create distributed circuits, you have to know that you are using something with specific rules. And I've been working in defining what a good architecture of chemical wire is, and based on the architecture, what does it predict? What is going to happen, right? And I've been trying to implement that with mixed results because of what you have been saying. It's very difficult sometimes to find positive results, not only because they disprove your hypothesis, not necessarily because of that, but because it's difficult for the experiments to work. So that's it. But the theory is there. I'm very proud of the theory.
My bachelor's thesis is basically, its aim is to engineer Escherichia coli, which is practically the default organism for research. And I'm trying to make it capable of surviving to the same environment that can be found at the exterior of the International Space Station. Because I read a paper on a cell that did survive, and I thought that, okay, we have cells that can survive, but can we actually engineer one to do that? And basically this means inducing resistances to a lot of things, including ionizing and UV radiation, a very wide range of temperatures, low pressures, microgravity, and it's being a total challenge. But I'm right now working on the radiation resistance, and it seems to be working more or less well.
In case I'm trying to develop a new photodynamic therapy. So, well, if you don't know what it is, the idea of photodynamic therapy is trying to use like an agent that is light sensitive and a light source, and then trying to use this agent and this light source in order to destroy abnormal cells. This is like a very brief summary. So what I am trying to use as an agent, I am trying to use nanoparticles. In this case, I'm testing what type of nanoparticles would be more good for this purpose. I'm testing some nanoparticles made of titania, also some that are made of titania with gold, etc. So I am like trying to develop a new therapy that would try to kill these abnormal cells that are there, and we have to kill them. And that's basically it.